If you have heat-sensitive items, it's a good idea to sterilize them with the filter. So here's an apparatus shown where you have you are putting your substance into an upper chamber right here. You're creating a vacuum, sucking the air out with a pump, and that will pull the liquid through the filter membrane into the lower chamber right there. And all the bacteria, they're supposed to be stuck here at the filter membrane. It has a fairly small cutoff, so most bacteria should really, if not all, should really get stuck here in this filter membrane. So the cutoff is usually on those membrane filters 0.22 micrometers. Here are some physical methods of microbial control. Something that we all do is refrigeration. So we're lowering the temperature, and that slows down the growth of bacteria. So it has a bacteriostatic effect, not a bactericidal effect. So keep that in mind. It's static. It slows down the growth. Refrigeration is good. Deep freezing is better. There you can store your items even longer without them spoiling. And then lyophilization, that's even better. That's freeze drying. So not only are you freezing your stuff, but you're also drying it out. High pressure, we talked already about that denatures proteins, especially high pressure steam. That's a really effective way to get rid of bacteria. And then desiccation, drying it out. In the absence of water, bacteria can't grow. It prevents their metabolism. They would not be able to multiply. Another good way to control microbial growth is applying a very high osmotic pressure on the outside. So if you want to pickle something, high salt concentrations will preserve your food items, or also high sugar concentrations like in jams and jellies. They actually work as a preservative just by creating this high osmotic pressure, and that basically sucks water out of cells and causes plasmolysis, and in that hypertonic environment, and it causes these bacteria to not be able to grow. Radiation is another great way to get rid of bacteria. Ionizing radiation, that's the harshest method here. X-rays, gamma rays, electron beams, they cause damage to the DNA that causes cells to die. A great way that's actually easy to use is non-ionizing radiation in the UV range. So that will be a 260 nanometer UV light, and that damages DNA by creating thymine dimers. And we have a lab on that showing the effectiveness of UV radiation. It's actually pretty amazing how effective UV light is. In hospital settings, a lot of rooms are exposed to UV radiation overnight to sterilize materials that are in the room. It works great. It's just the UV light has to actually reach the item. So if something is shielded or the UV light doesn't reach it, of course it can't kill bacteria. But anything where bacteria are exposed to UV light, 260 nanometers is ideal. Then the DNA damage, these thymine dimers, they prevent bacteria from being able to reproduce. Another way is the microwave, and that kills by heat. It heats up water molecules, and that heat denatures proteins, and that's how you can get rid of bacteria as well. So here we have the electromagnetic spectrum, and you can see sort of the visible light over here, and bactericidal, that range is down here in the UV range, and even shorter. So these the bactericidal range right here, 260 nanometers is great to get rid of bacteria. And even shorter wavelengths like gamma rays, they're even more effective. They're bactericidal. Over here in the very long range, there we have the microwaves. They're off scale right here, so they're just really long wavelengths, but they will kill by heat, not by damaging the DNA. Now let's talk about some principles of effective disinfection. Some things to consider would be the concentration of your disinfectant that you need to have in order to be effective, whether or not you are disinfecting organic matter. The pH needs to be considered a lot of times for effective disinfection, and the time that you need to apply your disinfectant to be effective. popular way of figuring out what's an effective concentration and time would be a use dilution test. And the way it works is you take a metal cylinder that's dipped in a bacterial culture, and then you're placing that cylinder in a disinfectant for 10 minutes at 20 degrees Celsius. Then you transfer that cylinder to culture media to determine whether there were any bacteria that have survived, and then whether your culture medium turns cloudy and shows bacterial growth. A better way, in my opinion, is the disc diffusion method, and that's something we would have done in the lab, where you are taking little filter paper discs and they're soaked in the chemical that you want to test out and see if it's effective as an antimicrobial agent. So you're dipping these sterilized filter paper discs into the solution you want to test, and then you're placing that soaked filter paper disc on a plate, on a Petri plate that has been swabbed with a certain bacterium that you want to test. And then you look for a zone of inhibition around the disc. So here's an example for that. You're evaluating disinfectants. So you're taking these little filter paper discs, these things right here, these filter paper discs. They were soaked, in this case here, in chlorine. And then you're putting them onto a Petri plate that has been swabbed out, here in this case, with Staphylococcus aureus. And so you can see that the chlorine is pretty effective. The O-phenol is not very effective. Quaternary ammonium, that's like a soap type of chemical, is not very effective here. And hexachlorophene is so-so. Chlorine is here the best. And let's take a look over here. Same chemicals are being tested here on E. coli, which is gram-negative, and you can see this is worse, actually. It's usually these disinfectants work actually better on gram-positives like Staph aureus, but here the chlorine is still pretty good. The O-phenol is not very good. Quaternary ammonium, not so good, and hexachlorophene doesn't really do anything at all. Here, one of the worst ones to get rid of is Pseudomonas aeruginosa, gram-negative, and this thing is notorious. It survives on your soap at home, and it's just hard to get rid of. So even chlorine is not doing much here, and none of the other chemicals seem to be doing anything at all. But that's a disc diffusion test, and it's a good way to test if your chemical that you think is has antimicrobial activity, you want to see if it actually does do that. Now let's take a look at groups of chemicals and how they might work as antimicrobial substances. A great way to disinfect anything would be phenols and phenolics, so similar to phenol kind of compounds. Problem with those is that they cause a lot of damage to our cells, and so if you want to wash your hands with phenols, not recommended, because it will cause problems with your own skin then. So phenols, they injure lipids or plasma membranes, causing leakage. It's great to get rid of bacteria, but again, these are very harsh chemicals that will cause a lot of damage to your skin too. And so here would be the basic chemical. Here is phenol over here, and then here's a phenol derivative. This is O-phenol that was on that this diffusion test that we had a couple slides back. Bisphenols, those are chemicals that contain two phenol groups, so that would be like this O-phenol. They're pretty popular as disinfectants, and there you have hexachlorophene, triclosan. They disrupt plasma membranes, and they're pretty popular as disinfectants, usually in hospital settings. So here would be two phenol rings, and there you have this bridge right here. So here would be hexachlorophene, it's a bisphenol, and then here triclosan, also very popular disinfectant in hospital settings. You can see, you don't need to memorize the formulas, but it's a good way to know what a bisphenol is. So two phenol rings, the phenol ring being this structure right here. Next up, the biguanides. Chlorhexidine, very popular substance that's used in surgical hand scrubs. It disrupts the plasma membranes of cells and is very effective as a disinfectant. Also very effective halogens, like iodine, comes a lot of times as a tincture, where you have an aqueous alcohol, the iodine dissolved, or you can have it as iodophor combined with organic molecules. It impairs protein synthesis, alters membranes, and thereby inhibits growth of bacteria. Then chlorine, I'm sure you've smelled it in swimming pools, is a popular way of keeping bacterial populations down. It's an oxidizing agent, shuts down cellular enzyme systems. If you really want to get rid of bacteria, bleach something. That would be hypochlorous acid, is very effective way. Any form of bleach is very effective. And so there, then also chloramine is chlorine plus ammonia. All of these chlorine-derived substances are very effective, but not the healthiest, to be honest. Other popular disinfectants include alcohols, and I'm sure you've used a rubbing alcohol. That would be isopropanol or ethanol. All of these are great. They denature proteins and dissolve lipids. They unfortunately do not kill endospores or non-envelope viruses, but other than that, if something has a phospholipid bilayer, then that should be affected by alcohols. So most popular would be ethanol and isopropanol. That's rubbing alcohol over here. And one thing to keep in mind is you're going to have to add some water, because if you don't, then all you're going to do is sort of the bacteria will survive, and they will be, after the alcohol is gone, then they will just regrow. So if you really want to kill something with alcohol or rubbing alcohol, then you need to make sure that there's at least a little bit of water in your alcohol. So here is a chart that shows you the effective concentrations of ethanol to kill Streptococcus pyogenes in this case. So you can take a look at this here. If you're using 100% ethanol, and it might be a surprise to some of you, if you're using 100%, you get no bactericidal effect, none whatsoever. And the reason for that is that the alcohol needs to dissolve or weaken the membrane, the cell wall of the bacterium, and then water needs to rush in and then lyse the cells, burst it basically. So, but if there's no water, then this process will not happen, and so you are going to have no effect. Now the concentrations here, anywhere from 95% alcohol down to 60. So you're cutting it kind of close here. That is effective. Most of the time, rubbing alcohol and alcohol that you can buy as a disinfectant, they're 70%. So you're pretty safe. You will find no growth at any of the times of exposure that are listed right here: 10, 20, 30, 40, 50 seconds. If your alcohol starts to evaporate, you're kind of starting to cut it close, because if you're falling below 60, you're going to end up having to incubate your items longer in that solution to achieve no growth, which is here NG, no growth. And if you're down to 40%, well, then your alcohol doesn't have any more effect. So if you think you can kill a bacteria with vodka, which is about, 40% ethanol, you are wrong. 40% is not going to do it. Next up are heavy metals and the effect that they have on bacterial growth. There's something called oligodynamic action. Oligo means a few, dynamic means powerful. So a small amount of heavy metals has a very large effect and has strong antimicrobial activity. Namely, silver and copper are great, and mercury, of course, has its toxicity. Zinc also works, but silver and copper are actually very popular metals that are used as antimicrobial agents, and we'll continue that in the next video.